pH meter video group members Erin, Jordan, Karissa, and Anabel.
After ensuring that the pH meter is securely plugged in, each group member carefully unscrews and removes the probe from the storage solution, ensuring that they do not slide the cap down on the probe. The probe is then rinsed with deionized water over a waste beaker and placed into another beaker of deionized water to prevent the probe from drying out if it is not being used immediately. The students then calibrate the pH meter using a 2-point calibration technique. The probe is first placed into a beaker of pH 7 buffer solution, and lab members swirl the probe gently in the solution until the value shown stabilizes. Then, using the stylus, group members enter the LabQuest program on the pH meter, navigate to the Calibrate Now function, enter a pH of 7 for the first buffer, and press Keep to save the value. The probe is then rinsed again with DI water, and the process is repeated using a pH 4 buffer, since acids are being tested in the experiment. For testing alkaline solutions, a pH 10 buffer would be used instead.
Measuring unknown solutions. Once the meter is calibrated, it is ready to be rinsed and used to test the unknown acidic solutions. Each group member calibrates the pH meter and tests one of the 4 unknown solutions, and the values are recorded in each member's lab notebook. The solutions tested were orange juice, apple juice, hydrochloric acid, and acetic acid, which were found to have pH values of 3.86, 3.59, 1.17, and 2.71, respectively. Finally, the pH probe is rinsed with DI water and placed back into the storage solution.
The pH of a solution at a certain concentration can be used to determine if the solution contains a strong or weak acid. This is due to the difference in proton dissociation between the two. A strong acid will dissociate completely in water, meaning that all of its molecules fall apart into hydronium cations and an anion. In a weak acid, however, only a small fraction of the molecules dissociate, meaning that there is a low concentration of hydronium ions in relation to a strong acid. If two solutions have the same concentration of acid, but one contains a strong acid and one contains a weak acid, the concentration of hydronium ions will be much higher and the pH will be much lower for the strong acid than for the solution of weak acid. This phenomenon makes sense when considering that the equation for pH is negative log concentration of hydronium ions. Since a strong acid dissociates completely, the rate constant for the reaction is so small that it is essentially negligible and does not really have an equilibrium. A weak acid, on the other hand, does reach an equilibrium. Since they do not ionize completely, weak acids will fall apart and reform until equilibrium is reached. The change in pH when a strong acid or a weak acid is added to a solution can be lessened by using a buffer to prevent fluctuations of the pH of the solution. A buffer solution, or one that prevents the pH of a solution from fluctuating when an acid or base is added, contains either a conjugate acid and a conjugate base, or a large amount of weak acid and a small amount of strong base. This buffer solution either donates or bonds with protons in solution to minimize the change in pH when a new solution is added by reducing the number of hydronium ions present.